Larvae of the small white cabbage butterfly, Pieris rapae, are a pest in agricultural settings. This caterpillar species feeds from plants in the cabbage family, which include many crops such as cabbage, broccoli, and Brussels sprouts, and many others. Interestingly, plants can evolve resistance to herbivores, such as Pieris rapae. And in this video, we will test the resistance of the model plant species Arabidopsis thaliana to this tissue-chewing insect using two methods. Hi, I'm Martin de Vos, a postdoctoral fellow in Georg Jander's lab at the Boyce Thompson Institute for Plant Research. Today I'd like to show you how to set up a choice and a no-choice experiment with Arabidopsis thaliana as a model plant, researching resistance against the pest Pieris rapae. Rearing of the caterpillars takes place on cabbage plants in the greenhouse. At least 2 cages are needed for the rearing of Pieris rapae. One for the larvae and the other contains the adults, the butterflies. In order to demonstrate the involvement of plant hormones and toxic plant chemicals in resistance to this insect pest, I'd like to demonstrate 2 experiments. The first will involve the determination of the role of jasmonic acid, a plant hormone often indicated in resistance to insects, in a no-choice experiment. Caterpillar growth can be compared on wild-type and mutant plants impaired in production of jasmonic acid. The hypothesis would be that larvae gain more weight